Welcome everyone to a new Expert Talk series. So glad you're with us. I got a little message for you because we need your help just like you need ours. As you're aware, research on ALS is continually progressing. And at Everything ALS, we are dedicated to uncovering new discoveries, aiding in early detection. As you are aware, research on ALS you can help us by participating in our speech study. The purpose of the speech study is to enhance the diagnosis and prognosis of ALS. We're seeking participants to provide 5 speech recordings, each lasting 15 minutes once a month, for a total of 75 minutes over 5 months. Very doable. We welcome healthy participants as well as individuals diagnosed or probable with ALS to join us. To enroll, it'll be in the chat also, but it's at everythingals.org/speech project. We also have one other study we are actively recruiting for, the asymptomatic gene carrier study. If you're a gene carrier and asymptomatic and interested in remotely monitoring your speech, cognition, and motor skills, please visit everythingals.org/asymptomatic study and fill out the form. We'll have that in the chat also. And thank you to each of you for being here tonight. And with that, I'll turn it over to you, Christian.
Thank you so much. Hi, everybody. Christian Rubio here. I'm glad to have you all join us again for another Expert Talk. And tonight, we're being joined by Dr. David Walk, MD, FAAN. He's professor and head of Neuromuscular Division at the Department of Neurology at the University of Minnesota. Dr. Walk is director of the ALS program at the University of Minnesota. He's the principal investigator of the FDA-funded clinic-based ALS data collection study and has extensive experience in ALS clinical research with a focus on data and biofluid collection and imaging. Dr. Walk's career focus has been on the diagnosis and management of neuropathy, including small fiber neuropathy, inflammatory neuropathy, and inherited neuropathy, as well as ALS and other motor neuron disorders. His clinic emphasizes multidisciplinary management, utilizing interdisciplinary teams of content and disease state experts. Dr. Walk received his MD at Brown University and did his residency in neurology at the University of Chicago and NYU. And he pursued his fellowship in clinical neurophysiology at the Michael Reese Hospital in Chicago, Illinois. We're really glad to have Dr. Walk. He's going to talk to us a lot about his natural history study, and the one that we are very proud to be collaborating with. And Dr. Walk, really the floor is yours. The stage is yours. Thank you for joining us.
Thank you very much. Appreciate the opportunity. I'm going to share, and we will get to work here. All right. Is the screen visible to all?
It is.
Yes, it is.
All right, great. So thank you. I'll be talking about what you see on the screen, a clinic-based, multi-center natural history and data and biofluid collection. And this started about a decade ago. The person who gets kind of the initial credit, in my view, is John Katz. Many of you know Dr. Katz, whose mantra was, we have all this information and we're not using it. And so several of us, Jimena Arcila, Dr. Arcila at Henry Ford Hospital, and I began this effort, which was kind of a bootstrap effort among several clinics and is now federally funded, to take advantage of the information that people share with us as part of their clinical care so that it might be used in ALS research. Now, this is the trick, is advancing the slides. Next. Aha, it worked. All right. And of course, since then, there's been a lot of work done with ALS TDI, everything ALS, now all ALS, create consortium, target, answer. And of course, one of the questions we get is, why this, are we all working across purposes or working collaboratively. But regardless, there's been a lot of work done that has recognized the value of learning more about this condition more broadly. And so the first question that I have here, the why, is why use ALS clinics for research? One doesn't have to do that. I mean, Everything ALS is a good example of how information could be obtained directly from people who are willing to share it for the purpose of learning more about this disease. There are certain benefits to using data collected in multidisciplinary clinics. First of all, as John Katz would say, it's already there. There's minimal burden and cost to use information that is routinely obtained for the purpose of clinical care, for the purpose of learning more about ALS. Second, it's inclusive. It's all people who are seen in our clinics have the opportunity to help us by allowing us to use de-identified information from their medical history. And not only those who are willing and able to come to a research office or are highly motivated to participate in a study from their home, or flying to a particular site for a deep dive. Our experience has been that about 90% of people who are approached with the question, would you be interested and willing to participate in a study in which we are using your de-identified data set to help us learn more about ALS more broadly? About 90% of people say yes. And what that means is that it's more inclusive than a self-selected subgroup that may be used to kind of model the disease and yet may not be as representative as something like this. Third reason is data quality. We're not perfect, nobody is, but the data are collected by trained and experienced staff, by individuals who work every week in an ALS clinic, are familiar and in our study have been trained to go through those questionnaires by physicians who have examined hundreds of thousands of people with this diagnosis. And any missing data is queried, and we are now having our data sets monitored as well. So I think there's a quality benefit to obtaining data from multidisciplinary ALS clinics. The whole story and the backstory. Our participants in this project agree to having data that precedes their diagnosis be shared, and data throughout their care for ALS be shared. So that provides the potential to do some epidemiological research, at least amongst the participants, as to what things have happened to them previously, as well as time to diagnosis, and what sort of testing has been done, necessary or unnecessary, prior to the diagnosis. And the data set, unlike, for example, a clinical trial-based data set, is not for a particular epoch of 6 months or 9 months or 12 months, but throughout the time that they are still being seen at that institution. And the backstory, again, is the other clinical information that may not even be part of kind of what we're collecting as part of somebody's ALS care. But there is the potential opportunity to look at serum creatinines and white blood cell counts for a period of time prior to the onset of symptoms or prior to the diagnosis. This is an opportunity to turn real-world data into real-world evidence, meaning that in the context of people who are being treated with a particular intervention, be it a medication, NIV, or something else, to then analyze potentially the impact of that intervention on their outcomes. Now, what we lack is the deep phenotyping that you can get from a study where somebody flies to Miami or Boston and spends several days being poked and prodded and assessed in multi-dimensional way. That provides an extraordinary amount of very, very valuable information, but requires a highly motivated subset and kind of a self-selection, if you will. So what we are doing is big picture, minimal barriers, broad inclusion. The depth of information that we obtain is based only upon the depth of information in a clinical record. So both ends of the spectrum have great value, but answer different questions. And I would argue that a clinic-based data set is actually not the end of a spectrum, but somewhere kind of in the middle. Because on the other end, we have nationwide or regional data sets that are collected by governments more effectively in Europe than in the US. But also, the CDC is a good example of how you can do that in the US. Those are broader. They're going to be yet more inclusive. We have one site in Philadelphia, so people who are going to the other clinics in Philadelphia are not included, to the extent that one site in Philadelphia might have a bias in terms of who goes there rather than elsewhere. We got a biased picture. On the other hand, I would argue we have far less bias than if you're looking at people who have the time, motivation, and resources to travel, for example. So again, a spectrum. We can learn a lot about the epidemiology of ALS from a national or regional or something or a data set based upon insurance information. We can learn a lot about ALS from highly motivated individuals who permit deep phenotyping, and we see ourselves as somewhere in between. So this is what we collect, and I've kind of split it up between person-specific and visit-specific. There are certain things that are not likely to change that we can obtain about an individual when they first enroll, regardless of when relative to their diagnosis they enroll. So the onset, the time of onset, the site of onset, their initial EMG and examination, the way that we characterize ALS, in terms of the ALS type. The genetics are not likely to change. The diagnostic delay is something that's important and isn't going to change. But then there are visit-specific pieces of information that tell us a lot about how a person's condition changes over time. Vital signs and weight, changes in medications, measures of function, and events, relevant events, general medical events, falls, blood clots, infections, and the like, and ALS-specific events, like the loss of certain functions. In addition, we are funded to collect blood, and we can do a lot with blood, plasma and serum. I didn't list it here, but we were having a conversation. There's a study that we're doing right now to try to develop a biomarker, a diagnostic biomarker, and for this particular thing, what's better, plasma or serum? We're collecting those. We're collecting DNA and doing whole genome sequencing in everybody who participates. That could be extraordinarily helpful as we're learning that there might be different normal patterns in the normal population unrelated to ALS, but that might present with different types of ALS and different potential personalized medicine options. And we're collecting cells. And of course, cells are important because they can be dedifferentiated and then redifferentiated and turned into neurons, or for that matter, glial cells, brain cells, spinal cord cells that are unique to that individual and might carry certain characteristics, maybe they genetic or otherwise. So these are things that are collected as part of this study. These are the current sets of investigators in the US. We are bringing on two that's close to being brought on. Well, they'll give us more geographic and likely more ethnic diversity than we currently have. But again, this is clinic-based. So in order for somebody to be a participant, they have to be seen at one of those institutions. And our data management is the NCRI in Boston, which provides certain benefits because of the overlap with many of the other ALS studies. And as many of you know, they've got individual-specific identifiers and then study-specific identifiers. So, I might have a global identifier for every study that I'm participating in, but a different identifier for this study from that one so that they can track who's who where is there duplication, and then where is there overlap, which again was what was referenced earlier. The hope is that we're not doing home digital data collection. So if somebody's participating through Everything ALS and happens to be participating in our study, we can marry the digital data sets that are obtained at home on a much more frequent basis with the kind of fundamental clinical examination data sets and historical medical data sets that are obtained in our study by identifying that these two different study-specific identifiers map to the same individual identifier. So what have we done? What's it been used for? I'm just giving you some examples because there have been many over the last decade that we've been doing this. First is just understanding ALS from a broad cohort. Again, to my argument that over 90% of people who are seen in 10 different clinics around the country is going to tell us more about ALS broadly than a self-selected group because they were in a clinical trial for a period of 6 months or because they volunteered for a deep phenotyping study. Top left is our publication that simply described the baseline characteristics of the individuals in the study, the rate of disease progression, the types of ALS, the ethnic and racial backgrounds, which more closely approximated the CDC prediction or extrapolation of the US ALS population than, say, PROACT database. Below that, Dr. Rosenfeld, Loma Linda, is looking at different phenotypes and the rate of change amongst different ALS phenotypes to better understand how this disease changes depending upon the type of ALS that you have. He's a strong believer that we're trying one-size-fits-all treatments when in fact, we're all different, different types of ALS, different phenotypes, and a group like this with up to 3,000 individuals is a perfect sort of longitudinal series study to make that argument. So he's used that to kind of dive deeper into the types of ALS and how that changes amongst individuals. Below that, we are looking at rates of change and time to events in different domains of ALS. On the right, I've got only three of, there's probably been 20 different posters from investigators in the consortium looking at specific questions. These are from Temple, looking at plateaus, healthcare disparities, and cognitive involvement. Others have looked at lipid metabolism within this cohort, comorbidities, and a variety of other things. So it's a rich data set from which one can ask a lot of questions about ALS and understanding it from a broad cohort that we think is more representative than some of the other great data sets that are available. Second, disease modeling. We're funded to do this, and we've begun doing it prior to our FDA funding. In the top right, Ernest Frankel at MIT described and built models for how ALS progresses differently in different individuals and used our data set as well as PROACT, as well as the Emory data set to build those models. Top right is our first publication on time to event prediction. So this is an actual prediction model. The bottom are other more sophisticated prediction models that we're creating as well. The concept behind prediction, as you can imagine, is for an individual, it can help plan and recognize where things are headed, and it can be updated with each visit. For a population, it can be used to develop clinical trials. So trialists want to know how best to show a potential benefit of a treatment in the shortest period of time, the highest likelihood of success, and in the right population of individuals. So having a clear understanding of what group do we want in our study, or conversely, of those in our study, what can we learn about the likelihood of reaching a certain point with or without our treatment helps power those studies. So it's very, very valuable, and this is the kind of work that when studies are being designed, clinical trials are being designed, is looked at. I also personally feel that there could be benefit in using time to event as an outcome. And there's been a lot of conversation about this. And fundamentally, ultimately, for a registration study, a phase 3 study, one has to use outcomes that the FDA has shown to be validated and fit for purpose. But we've all had conversations about the ALSFRS-R and its strengths and shortcomings, and its clinical relevance or lack of clinical relevance. Time to events that matter in people's lives, the loss of the ability to perform a certain important function might be an outcome that can be very important and very useful in trying to determine if something is helping, because those are events that happen during a person's lifetime as opposed to survival, which is of critical importance, but is only a single event that we hope is not triggered during the course of a person's participation in a clinical trial. So these are some of the things that we and others are doing to use this information for modeling ALS. Another example is evaluating treatments. And again, everybody on this call is very familiar and have heard conversations about propensity score matching, virtual controls, a variety of statistical approaches. But in order to build those approaches, whichever one uses, one needs to know something about the condition itself. So I'm showing just one example that MT Pharma has used in trying to get a better understanding of whether their drug is working. Obviously, they've got a proprietary interest in proving a point, but they're not the only ones who are utilizing data sets like ours as outcome measures. So another example would be in expanded access programs. Expanded access programs principally are for the purpose of providing a potentially effective treatment to people who otherwise are not able to participate in a clinical trial because of exclusion criteria, and providing the sponsor more safety data. But you can, as you can imagine, this and other data sets are being used to try to get a hint, a clue about the effectiveness of an intervention in expanded access. Certainly, if you've developed a drug and you're hoping it's effective, you are going to want to ask that question in the expanded access program. Can we demonstrate? Is there any way we can get a clue as to how this is working? Well, how are expanded access programs different? They're different in that the inclusion is much, much broader, in that you're involving people who often are farther along in their course of ALS, and in that there's a very broad range of individuals participating. How are you going to demonstrate that a treatment may or may not be effective with all of those kind of statistical hurdles? Well, one way to try is to have a data set of thousands of people, large enough that you can match the people in your expanded access program with people who might have similar characteristics and have been through this experience before. I'm not saying this is simple. I am not a biostatistician, and even for biostatisticians, one can imagine the complexity. And one of the most important ones is if you are comparing, Mr. Smith in 2025 with Mr. Jones in 2005, the care in ALS has changed over that period of time. Or Mr. Smith, who's highly motivated and participates in an EAP in 2025, with Mr. Jones, who was focused principally on quality of life and less on interventions in 2025, you have to recognize those differences and adjust for them. But again, this is a data set in which we have a lot of useful information that can help when people are developing these models identify the covariates that might allow us to match people as well as possible. So that's another utility of a large, ongoing, living data set. And then finally, we are just beginning to put the blood samples to work. I mentioned today we were working on a diagnostic biomarker. We are working on some progression biomarkers, and I didn't include it here, but predictive biomarkers that might tell us more about where ALS is headed. Now, NFL has worked pretty well in that domain, not as well as a biomarker of disease progression, or certainly, in my view, not as a diagnostic biomarker. But our blood collection, we are now using to develop these tools, and a bucket of blood is of no value unless it's anchored to a rich clinical data set. And internally, for some of the studies we're doing at the University of Minnesota, and externally for some of those who have approached us for biomarker studies, one of the appeals is that if you know a lot about the characteristics of the individuals from whom that blood came, the type of ALS they had, the rate of progression, and other general medical features that are part of the data set, then you learn a lot more about what that blood is telling you. And that goes to subtyping. Again, it may well be that some of the best treatments that are being developed are going to be best for certain individuals who might have a certain proteomic signature, which we can identify in their blood, or genomic signature. Amazing work is being done on this, which I was just learned about from a presentation a few months ago. We'll have those signatures as well as the clinical information. And if you don't put those things together, you really can't unlock some of the clues. It's kind of a Rosetta Stone sort of circumstance. So that's a fourth, I think, category in which data sets like this can be very helpful. And again, we're not the only one, but we're hoping that collectively we can all contribute to learning more about this disease and accelerate the work that's needed to advance treatments. So I believe that was the last slide. And if I'm correct, looks like I am, I'm going to stop sharing. And I appreciate the opportunity to have this discussion.
Thank you so much, David. We've got a few questions just to get the conversation going. And one in particular, there's been some questions about being a gene carrier. And one in particular was that there was one participant in the audience who said, I'm an ATNX2 gene carrier. I've never known or had heard if doctors are working on this gene. How can I find out? And I think there's a more general question as well. How can I find out and obtain more information about which genes are being worked on?
Yeah, that's a broad question. As you know, there was one study intervening in ataxin-2, and that's a complicated gene in a sense, in that its impact on ALS is a little different than many of the others. In terms of knowing where the work is being done, obviously, again, clinical trials, observational or interventional, are listed in clinicaltrials.gov. I believe, does Everything ALS also have, I believe you guys are also listing major trials. There are great sites to learn about what is being done. But I know more work is being done with ataxin-2. In terms of the genetic components, yeah, Dr. Ostro is going to have some answers here, I think. In terms of the genetic, what we're doing, we are a relatively small contributor compared, for example, to the Dutch group, but just starting to contribute genomes to the broader totality of genomic information, which I think is going to be, already has been critical in understanding ALS. But it may be that Dr. Ostro has some comments as well. I see his hand up. Or I see actually a yellow, that's not your hand. It's a weird avatar of a human hand.
I was actually going to ask a question, but
Okay.
but it does kind of relate to this. And so, David, I'm like a huge fan, right? But I'm also biased because I'm that Philadelphia site that you mentioned, right? So, but you started off talking about at the beginning about how important it is to have the rigorous clinical data, right? And that things like biological samples, really need that data to be informative. And we've talked a bunch about so Everything ALS has all this unbelievable digital data, and it's like a no-brainer to think about all the cool ways that you could work. Like you were just mentioning, figuring out subsets of people living with the disease. Is there certain things about the clinical data that clusters with things in biological fluids, but also with all this unbelievably robust digital work? And we've been trying kind of seat of our pants, right? Guerrilla warfare way to figure out how to link up the digital data. And can you just sort of talk a bit about, like, what are the barriers, and what do you think, and how can we, because it seems like when you're discussing things such as, identifying patterns, whether it's in different geographic locations or different types of ALS, that this is an instance that really the two types of information could really help. And just last part about that is, as far as the genetics go, I mean, obviously that's the next part of it, right? Because if there was a way to annotate digital information and clinical information and all of that together, well, then it's sort of this massive thing that we can look at to test hypotheses and figure out, who should get which trials, which drugs.
Yeah, I mean, the short answer to your first question is that there are some absurd kind of technical aspects in terms of connecting that we will get past. But the corollary is that anybody who is being seen at one of these centers, we want to be sure they know about the Everything ALS digital studies so that they can participate and so that we can link. And of course, just by virtue of the size of both, there are a handful of individuals who are in both, but we want that to be way more than a handful. But you're absolutely right. And there are those who have argued that with digital DHTs, maybe we don't need some of that clinical information as much. I believe we still do, because I think that there are very important pieces of information that we're collecting clinically that matter. Although I think that's still a big question. We know that, for example, there's clearly a distinction between bibrachial palsy, an individual with, weak, rapidly progressively weak flaccid arms and relative preserved function elsewhere, and a pure bulbar form of the disease, and a kind of more characteristic form of ALS. There are clear differences there, and there are probably genetic differences, gender differences, etc. Now, for three people in the same family, one of whom has bulbar onset and another has limb onset, is there a difference there? I don't know, but I still, I think this idea that different phenotypes, that is, different clinical features, might be telling us something is something that we need to take out for a spin. I've got certain skepticism about it, but not entirely. And so, yeah, we need a lot of that clinical information.
So very basically, so can you sort of comment on, so why does it matter? And what I mean by that is, so you mentioned sort of, disease models, and I think, just for the benefit of people, like, what is a disease model, and how is this sort of effort going to figure out who should get which medicines, right? Like, so in a very basic sense, this information and figuring out that, like, what's the next step? How does this help us figure out how to treat different people, or know what's going on in a model?
So this is a chat. There are people with far more expertise on all of this, right, than I. But I think there's a couple of things. Modeling, as I said before, I think modeling the progression of ALS has particular utility in terms of biostatistics and clinical trial design. And whether that's designing a large trial and making sure that you're maximizing the likelihood of showing a benefit if there is one, or whether that's asking a kind of upfront, how do I want to say this, like a futility design in a pragmatic trial, where you say, we don't have a ton of money for this study. We've got this really cool compound, and we want to find out quickly and inexpensively, and without putting anybody on placebo, whether there is anything to pursue here. Models can help with that, because if you can argue that if you can model the 200 people that are taking this drug, nobody on placebo, and you can model in advance what difference between the outcomes you anticipated and the outcomes you see would be sufficient to say this has a chance or this doesn't have a chance. You can move through compounds faster, especially some of the repurposing. Now, in terms of who should be on which drug, I think that that's where we need something more sophisticated. That's where we need the genomics, the proteomics. We need to understand who has perhaps a kind of large inflammatory component to their disease and who doesn't. Who has alleles, again, that might point them towards different processes damaging their cells.
I would offer also, some of the insights we get from our community too is there are things that we know they experience that aren't maybe considered part of that official disease modeling of ALS today, like pain or earlier phase type experiences that are so important for us to capture. I know we're trying to capture that with our app and in digital health technologies to get earlier definitions of the disease potentially earlier than how it's understood or even just really recognized for treatment today. And that can really fundamentally change a trial's design, right?
Right, and that goes to an advantage of what you're doing and a disadvantage of what we're doing. So whenever we, present this project, smart people say, well, you should capture this too, and you should capture that too. And I try to explain that we got to have a balance here because the whole idea behind what we're doing is keep it simple so that there's broad inclusion. Because the more complex you make a even if it's just data collection, even if it's just questionnaires, not blood or spinal fluid, the more complex you make it, the fewer people are going to participate. So that's why we need something like what you're doing, where you can easily ask and the participants can offer, hey, you should be asking us this. You can easily identify things that we don't have the time to do, and then bring that to the fore and say, yeah, pain matters, either in terms of as a quality of life issue or maybe there's a particular aspect that tells us something about pathology.
Yeah, I'm gonna weave in one of the questions I was asked too, because when it comes to pathology and understanding the disease, what if there's so many different what ifs with this disease? What if the disease has no connection with specific known genes? How can participating in a natural history study help at that point?
Well, again, this is not a genetic study solely. This is about ALS and everything that we define as ALS. And I should add, by the way, that includes people at genetic risk, although we've worked with all ALS to be sure we're not doubling up, that we're using the resources appropriately. But our inclusion criteria include PLS, people at genetic risk for ALS, in fact, include Kennedy's disease, other motor neuron disorders. So, obviously Kennedy's disease is genetic. Most ALS, we think, is not at least monogenic genetic. So this is important information regardless of the genetics. But I did mention the sequencing because I do think a lot is being learned, again, about the kind of, I'm going to use air quotes, normal variations in important genes that are relevant to disease. So that, I might have 311 genes that are somewhat protective and 417 that increase my risk of this condition or that, or that might make it more likely that if I do develop ALS, it's going to be lower motor neuron predominant and you can attack it this way or that. So genetics becomes more and more important every year, even if we say somebody has a sporadic disease.
Yeah.
I mean, I think that's true in just about everything except, murder by gunfire. The genetics probably doesn't matter there. But just about every acquired disease, the role of our genetic susceptibilities we're only beginning to understand.
Thank you. I'm going to go to the next question. Is it possible that a natural history data set might be more likely to address a question or a specific set of questions, e.g. as to why there's a higher rate of ALS occurrence in the state of Michigan? I think that speaks to the kinds of data being collected and how it's being kind of spliced and diced depending on the protocols and the aims, right?
Yeah, I think we're probably less good at that than other epidemiological surveys, like what the CDC is doing. And of course, there's somebody in Michigan who can speak to that much better than I, which is Steve Geltman. But we have the disadvantage of being specific to individual clinics.
Yeah. And by the way, for anybody, if that question was pointed towards Michigan, we do have a recent program that Dr. Steve Goutman gave, I believe, last summer, in which he went in depth in his study in Michigan. So worth watching on YouTube or in the app. I'll go on to the next question. Do you have any experience with this? Is for probably for any physicians on the call, including you, Dr. Walk. Do you have any experience with transcranial static magnetic stimulation for ALS? I believe there is an active study in that, but yeah, it's been used in Alzheimer's elsewhere.
I know that there is. I don't personally have experience of, enrolling folks in those studies or in doing that. I don't know if others on the call do.
Not at this time. Okay. And how many patients are in the database now, and how large would you like it to grow?
I believe so that we have data on approaching 3,000 individuals. There are about, I believe, 11. This has been going on for 10 years. I believe there's 1,100 and change who are currently contributing data to the data set. We are adding a few sites a year, and it's a little bit of a complex answer because what I am leading is, what I'm fortunate to have received is FDA funding, and there are some sites that have been brought on that are not part of the FDA project, but we're adding a couple per year, and as long as we still have the money to fund those sites, we're going to keep bringing them on to get the best numbers we can.
Thank you. There were a couple of questions about accessing medicines or treatments. Is it possible to get any trial drugs outside the U.S., specifically Turkey these days, or if there's any update on international collaborations and given all the changes in the world right now?
Others might have their own answers. I know very little about access in Turkey or in much of the rest of the world. I will say that I have not seen a change in the collaborative spirit of clinicians and scientists, and there are some barriers, but let's hope that we can continue to move forward.
Thank you. Is there any more information? I just saw a new one come up. Is there any more information on treatments for Qalsody and SOD1 or tofersen? I assume if that means of any expanded use of it, or
right. I mean, to my knowledge, it is still only being used in individuals with SOD1 variants, although I think there's also a certain amount of, there are certain variants where there's still some controversy about pathogenicity. I do know that there are those who feel that there may be a role for SOD1 in certain other individuals or more broadly in ALS, but I don't know how compelling that information is. Lyle, I don't know if you've got some thoughts about that.
I'm not sure what the question's specifically about, but I think that the real world evidence with Qalsody for people who have SOD1 mutations is continuing to be remarkably good, right? So there's people whose disease is stabilizing and really doing quite well. There have been some recent instances. There's a rare times where there are these inflammatory and autoimmune reactions, like idiosyncratic to Qalsody. Some people have gotten something called aseptic meningitis or myelitis, which is an inflammation of the spinal cord. These are treatable. They're treatable with things like steroids, but they're very serious. And it is something, although rare, that's happened, in a few cases. And I know there's been some recent work trying to kind of pool, how do we handle that? For example, do we treat through it? Do we give steroids to people that happens to and keep them on the drug, or does that mean they can't get the drug? I think that's evolving, but the experience with Qalsody as a treatment for SOD1, I think, is so far better than people maybe expected from the clinical trial data. So there's a lot of enthusiasm.
Yeah, and it just goes again to I make a big point at the time of diagnosis that it's critical that people have undergo genetic testing because in addition to that, there are, emerging or experimental antisense oligonucleotides for other rare ALS genetic forms, and it's kind of a direct kind of targeted backdoor to potentially effective therapy for those individuals. We don't want to miss anybody who might be a candidate for antisense therapy, whether it's the one FDA approved or others that might be coming along.
The last thing I'll say about it too, I think that a lot of people in the chat have been mentioning and the legacy, which is this unbelievable group of people doing all sorts of stuff for genetic ALS. But in particular, we have this growing population of people who, what we call at risk, right? So people who have a genetic mutation such as SOD1 that we know causes ALS, but they haven't manifested ALS. And the reason we have a growing population is because of what you just said. Everybody's obviously who has ALS gets tested. And then if they get tested, maybe their family members get tested. And personally, one of the things I'm most excited for the digital tech, like everything ALS is doing, and in combination with this natural history study that you're doing, is, we got to learn how to do trials in people who haven't manifested disease yet, who have these mutations. We have to figure out what should we be tracking, because we don't have the bandwidth to bring them into our clinics and see them routinely. And so what is the important kinds of data? What is the important kinds of clinical information that we're going to be able to follow? Because someday, we want to be able to give these medicines before somebody has ALS.
Right. That's a really, really valuable point. I'm just thinking, just today I saw somebody who's we've been following, just, comes in periodically. I do a neurologic examination, not a very sensitive way of judging, and, do NFLs, but I do believe we're going to get to the point where not just a kind of a pre-symptomatic rise in NFL, but we're going to have highly sensitive, minimally or non-invasive ways of identifying that TDP-43 aggregates are present somewhere and are growing. I think we're gonna get there. But DHTs, you're right, are a great way to do that. And we're all, where's mine? We've all got one on us. And so anybody in that circumstance, if they just kind of run their cell phone calls, through an analyzer every 6 months or so, you've got one right there, whether you're in a study or not.
I mean, there's people talking about SOD1. I think the estimate is that there's 300 people with SOD1 ALS or so in this country, something like that. And it just points out, I saw Lynn put in the chat that he's done like 11 million speech samples, which I know there's several people on this call right now. You guys are all amazing warriors who do everything and involve yourselves in all these studies, and just realize how valuable your data is, right? And, it's just unbelievable. It is so important to this field for people to be participating in natural history in this study. I mean, how are we going to figure this out with these small numbers? So yeah, I mean, we were joking with Lynn, we need a leaderboard. I think, Christian, we do need a leaderboard. Like people who participate in the most, we should have some sort of.
I put a little footnote in my own head, like I'm making a note. Product feature. I'll add also from my times on the pharma side. I know people from the community know I've worked on the pharma side. This is all a community chess match too, of unlocking a disease and getting forward. Nobody's a pawn, everybody's a player, and the pieces in front of you include participating in studies where we're unearthing completely new data. So to me, when I think about the role of these digital health technologies in a space like an asymptomatic gene carrier, it's so important because from the pharma side, they can't make the case with data that doesn't exist for new people to potentially get access to tofersen if they don't have any symptoms. But when an asymptomatic study shows that maybe symptoms are happening earlier, and you can really build a case around that, and then you layer that with a natural history study, now we've collectively moved that goalpost earlier, and now it really unlocks a lot more access to people. And that's just one. That's just for SOD1. There are, like you said, Dr. Walk, there are many more coming to break this disease down into its many potential unique individual facets. So that's why it's so important for people to see the opportunity to help be a part of creating these subpopulations that kind of make the goal bigger for companies to work on, researchers and drug developers to work on.
Yeah, when we have the targeted treatment for C9, we will need to figure out how to follow people and when to start treatment. And those actually aren't simple questions, you know.
I think it's a great question from Eric. Also, we had a question, I appreciate the efforts that try to find it earlier. What about folks who have been around 5 years now? What needs to be tested? I think, yeah, go ahead.
Oh, no, if the question is if they've not had genetic testing, it's never too, I guess I shouldn't say it's never too late, but it's, that should be done. But I'm not, that's what Eric's asking.
I wonder also if what else can be done for folks who've had onset for years now. They're still around, still fighting. And what can we do with these natural history studies and trial designs to be more inclusive of them?
Right, and I think that's again, EAPs are an example of that, because some of those individuals will be in EAPs, and it's also the case that again, there are ways to impact somebody's care. We haven't even talked about care. We've been talking about interventions that are intended to kind of modify the disease, but there are ways to impact somebody's care regardless of the duration of the disease. And one of the things that I find challenging sometimes as a clinician is when you get to know somebody over a matter of 5 or 10 years or more in clinic, it's very easy to kind of get fooled into thinking that things aren't moving much when in fact they are. And you kind of, if you're not looking carefully at their examination or their symptoms or their scores, you can get lulled. And so if we have things that even have a slow and modest benefit, if somebody's had the disease for 5 to 10 years and we can keep them walking for an extra 2 years instead of an extra 6 months, that's a real thing.
I actually think of it maybe the opposite way also. So what I mean by that, I think that the value of collecting all of this data. So in other diseases like HIV, for example, or traumatic brain injury, they learned a lot by studying the people who progressed slowly, right? The people who had HIV for decades and never developed AIDS.
Yeah.
The people who were the sparring partners, boxers, and got punched in the head for years and years and never got traumatic brain injury, right? And what was different about them? And in my mind, efforts like natural history, efforts that are collecting clinical information, the super valuable people in those, and
yeah.
digital efforts to me are those people, because I want to know what's different and is there something targetable. And I think just this week, we were talking about gene testing. I saw an article, I don't remember the gene right now, but there's a gene that's just been discovered that may in fact be protective, right? That may be a particular gene that people who have slowly progressing ALS have, a particular mutation. And so that gives us an idea of maybe a new therapy to target. And the only way we would find that out is if we were collecting information on these people who have very slow, long ALS in a rigorous and coordinated way.
Right, and a good example of that, we're making neurons from people with PLS and people with ALS because of the possibility that we're making spinal motor neurons to see if they have some sort of protective factors that people with ALS should have.
We got a question.
Raised hand.
We've got another virtual hand up from Wolfgang.
Yeah, so that echoes what my good colleague and friend Richard Bedle is doing at Duke University in his ALS clinics, where people come to see him, a lot of people. I know because I was once a colleague of his working alongside him. I'm not an ALS neuromuscular physician. I was treating patients with chronic pain, but working alongside Rick and also receiving his referrals. And brainstorming with him about the disease. And I thought what he's doing is very, very valuable to be a connection, collecting basket for this slow ALS long-term, and also what, with quotation mark, ALS reversal. And I think, Lyle, what you were referring to is one of Rick's first publications on this cohort, identifying a protein that's involved in IGF receptor signaling, or gene that's involved in IGF receptor signaling that stands out as as being different in the cohort of so-called ALS reversals, and that's the first stone that is coming out of the water. And so there will be more, I'd say. I have one comment on that clinical course, that there are people with long-term course, and then if you can prolong their being mobile and being able, to walk for 2 more years. So for that subgroup of ALS, I think it's going in a direction that it will be neuroprotective and sort of like neuroregenerative treatments, which will have to be launched and have to be refocused on for that purpose. Whereas if you have those patients where you want to intervene super early, perhaps like pre-symptomatic, pre-oligosymptomatic, that will be likely going in a direction to interrupt disease mechanisms that are identified for the specific cause. So it perhaps, crystal balling, it might become a personalized medicine spiel. And then for those down the line who are longer term suffering, it will become something that is shared with other diseases where neurons go down. And it could be neurodegeneration, but it could also be borrowing from the textbook of neuroprotection in stroke, catchword edaravone, and in combination of edaravone with other neuroprotective agents that might help us out in this particular and in that section of the battlefield. But I'll stop there.
It actually, so I love that study you mentioned from Dr. Bedle. I was actually talking about a different one. I think the proteins, I just was trying to find it right now. I think it's Krebs3, completely unrelated to what Dr. Bedle's doing, done by Dr. Brian Traynor and some other geneticists. They just this week have found this variant. But mentioning what Dr. Bedle has been doing, and you're right, those are super exciting studies. There's another part of that. I don't know if you've seen, he had some work that he presented about microbiome and about different gut bacteria associated with slow and fast ALS, and actually they presented something in NEOS where they took stool samples from people with fast ALS and slow ALS and gave them to mice and were able to speed up and slow down ALS based on something in the stool. And the reason I mention it is, this is one of the things that makes me excited about, for example, the Everything ALS nutrition study, right? The app study, because we have some evidence that things like gut microbiome and maybe diet are doing things that we don't understand, either to slow down or speed up the disease. So, but the one I was mentioning, it's completely separate from Dr. Bedle. I think it's called Krebs3 if somebody wants to Google it. Anyway.
Well, to vindicate Dr. Bedle, he might find that as he continues to analyze, because this is just the first one that he hit on and that he has shared with the world. And if you screen for genetic mutations that do something, you discover the first one, and then it becomes like a mosaic that tells you, okay, the biology goes like da 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 da. And so ALS reversal, ALS slow clinical course, that spiel in terms of genetics is just starting.
We were close to that time, but thank you for everybody for that for the robust conversation. Dr. Walk, thank you for the presentation on the natural history study, and thank you for launching something that has so many different institutions and the network growing. And we really appreciate the opportunity to collaborate with you and everybody in this network of this study to help generate not only the foot traffic into the door to get to participate to the core of the study, but also complementary data that can really help advance the field. So thank you again.
Thank you for the opportunity to join you.
Thank you, Dr. Ostro, and everybody else who's joined the conversation as well. Thanks to everybody who asked your questions as well. So with that, McFinn, I'm going to turn it on back to you.
All right, everybody. There you have it in your living room. Came right to you. This is the opportunity to find out things that maybe you've never heard about, but your questions are so important. Thank you for being so inquisitive, because without you, we wouldn't think of some of these things.